Yeah.
And I am recording. I got all this stuff I got to make sure is running right. Okay, so we are now entering into our biochemistry side of this course. And we're beginning tonight with carbohydrates. This is a major source of energy for the body, and basically everything surrounds carbohydrates. And carbs can be found in all kinds of foods, fruits, legumes, breads, and all kinds of grains. We've got rice over here and oats. I guess that's probably what that is. Potatoes, vegetables, all kinds of things have carbohydrates in them. Oh, these are, I guess these are beans too. So if you look at biochemical substances, the whole world of biochemical substances, you have inorganic biochemical substances and then bioorganic substances. Oh, we lost somebody coming in now. Um, this is kind of the makeup of the human body. So you can see here that about 70% is an inorganic, is just water. About 5% are inorganic salts. About 15% are proteins. 8% are lipids. We'll be talking about these in the upcoming weeks. Carbohydrates is about 2%. Nucleic acids about 2%. So about 75% of the plant biomass are carbohydrates. So if you weighed all the plants in the world, about 75% of that weight is due to carbohydrates. So plants are a major source of carbohydrates, as we just saw. They are produced through photosynthesis. For humans, carbs pretty much drive us. We're carb machines. It provides energy. It provides short-term energy storage as glycogens. We'll get into what this is in a little bit. Carbs become the supply carbon for us to synthesize proteins and lipids and so forth in our bodies. It also provides part of what makes up our DNA and RNA, which we will get into as well. In conjunction with lipids, they form the components of cell membranes. So the cell membrane is what keeps your cell intact and separate from other cells in the body, and this is made up primarily of carbohydrates and lipids. In conjunction with proteins, they function between cells and between cells and molecules in some recognition processes. So carbohydrates function as like a flag that will tell cells what this item is and how to interact with it. Oh, bounced out again, huh? Let's make sure everybody's here. So we will get into all of this in the upcoming few weeks. Okay, carbohydrates are polyhydroxyaldehydes and also polyhydroxyketones. So you see over here, this is a carbohydrate called glucose, an extremely common one. And you see here that it's a long chain with a bunch of hydroxy groups on it. At this end is an alcohol. And up here we have an aldehyde group. So this is a polyhydroxyaldehyde. Fructose, a common component of in fruits, you have a ketone group rather than an aldehyde group. And quite often you end up with an alcohol on either end like this, but here's a ketone group. And then of course you have the polyhydroxy groups in here as well. They can exist as linear chains, just as we've shown here, but they are more stable in cyclic structures and often exist this way. So what can happen is this end can end up connecting to this end in order, this is glucose in a ring formation, and it's more stable than being in a straight chain. And here's fructose where the ends have combined with each other, and this is a fructose in the structure. There are different types of carbohydrates. So monosaccharides is a single unit of either a polyhydroxyaldehyde, these are called aldoses, or polyhydroxy ketones, those are called ketoses. So a monosaccharide is just going to be one of those. So here is galactose, and this would be a monosaccharide. If you have 2 to 10 of these, however, chained together, so 2 to 10 different of these carbohydrates chained together, these are called oligosaccharides. And oligo kind of means a group, like an oligarchy is a country led by a group of people. That's where this oligo comes from. So example of this is common table sugar. Sucrose is an oligosaccharide, and it is a glucose connected to a fructose. So here we have a glucose molecule, and then it's bridged together with what's called a glycosidic link to the fructose. And we'll talk about this more in a minute. But so this is an example of a oligosaccharide. Polysaccharides are going to be 20 or more monosaccharide units. And so again, it's like a polymer, just like we've seen over and over again. You've got this linkage here between one monosaccharide and the next. And that this, just like with our other polymers, is repeating a n number of times and making these long chains. Examples of this are cellulose and starch. And we'll be looking at those in a minute as well.
What would something be called if it's between
10
11
and 20.
19?
I was waiting for somebody to ask that. Yeah, I don't know why they do that. I guess, for purpose of this class, I guess just think of this as being greater than 10. It's not that big of a deal, but that's how they define them. Kind of strange they would do that. Maybe there is a separate term. I'd have to go back and look at that, but I don't believe there is. We could make one up. Okay, monosaccharides are usually named for the functional group and the number of carbons in the chain. So here we have an aldehyde on this end. So this is called an aldopentose because we've got 1, 2, 3, 4, 5 carbons. So the aldo for the aldehyde, and then the pent for 5 gives us aldopentose. Here we have 6 carbons in the chain, but it's got a ketone group in it, this carbonyl group right here. Remember that could be on the test, carbonyl group. And so this is called a ketohexose. Keto for this carbonyl ketone group here, and hexose for the 6 carbons in the chain. So these are like category names, so to speak. All right, chirality. Have you guys done chirality in lab? I know you guys have missed some
We just
labs.
did.
You just did
That
it?
last lab we did.
Oh, okay. So that you didn't get snowed out from that one? So I'll run through this, but if you've already done it, this will probably be a little bit of review for you. But chirality is a molecule or an ion that cannot be superimposed on its mirror image of itself. The way to think about this, chirality is literally comes from the Greek word for hand. Cher means hand. I had to put this in here because I love languages. And so our hands are an example of this. You probably did this in the lab. Our hands are mirror images of each other, and there's nothing you can do to make these superimpose on each other. You can even put them together, but these are still not identical because your palm is facing this way and this palm is facing that way. So they're still not truly superimposed. So our hands are a good example of chirality, and that's literally where the word comes from. Um, so chiral objects are like the handedness that we talked about. Achiral objects can be superimposed on each other. So here we have two Erlenmeyer flask, and they're a mirror image, but they can also just be put on top of each other and they match up. So basically, it's if the item has a plane of symmetry in it, it's probably going to be achiral. Here you don't have any plane of symmetry. If I go down the middle of the hand this way, it's not a mirror image because there's no thumb over here. If I go this way, there's no mirror image because the fingers are up here and the palm is down there. So this is going to be a chiral object. A chiral molecule, for purposes of this class, we're going to be focused on carbon. And the general rule that we're going to use is that basically for carbon to be chiral, it has to be bonded to 4 different objects, 4 different things. So here we have an example of a carbon that's got a chlorine, a hydrogen, a fluorine, and a bromine. If we make a chemical compound that's identical to this, but a mirror image, so this, if you drew a mirror down the middle, this is a mirror image of that. These are isomers because they have the exact same chemical composition, but they have a different orientation in space. And there's no way that you could put this superimposed on that and make them match up. Do you guys want to step through that, or you guys are like, this is all review, we don't care? What do you say? You guys feel like you got this down pretty good, or you're good? Okay. I do have a video here if you want to watch it, if you're still kind of struggling with that idea. Ziggy Plant on. Ziggy Plant, thank you. So there is a video here that kind of explains chirality. So if you want to look at that, we won't watch it tonight. And did you guys talk about why this is important? That chiral chemicals will interact with light in different ways. And this is called optical rotation. So if you take a plain polarized light and pass it through a solution of chiral molecules, it will rotate that light a certain degree, either right or left, depending on this object's orientation. And the reason for this is that light has some element of chirality to it as well. So when it interacts with this molecule, part of that light will come out at a different speed than another part of that light, and it will rotate it one way or the other. So chiral centers then are carbons that have 4 different groups associated with them. So here we have a carbon, and it's got a hydrogen here, it's got a hydroxy group here, and then here it's got a CH2OH. So that's different than either one of these. And here we've got a CH double bond O. So this is different than this, or that. So this is a chiral carbon, and this is a chiral center. Any questions there? I don't know why I have this up here. Oh, no. This is the same thing as that, so I don't know why I have this up here. If you put a hydrogen in here, this would no longer be chiral. This carbon would no longer be chiral. This carbon is not chiral, and why is that? Anybody?
It's not bound to multiple different things. It's bound to another carbon.
Well, yeah, it's bound to an OH, and it's bounded to a carbon, and then it's got to then have 2 H's here, right? That's implied in the stick drawing. So you're exactly right. Because I've got 2 items that are the same, this is not going to be chiral. And here's an example. I don't know why I have this here either, but this is an example of D-glyceraldehyde. And this is an example of L-glyceraldehyde. This one will, because we'll get into this in a minute, but D rotates light to the right and L rotates light to the right. Can't talk today. Light to the left. And this is a chiral carbon again, because it's got 4 different groups to it. And this is called a Fisher projection. We're going to look at that. Did you guys do Fisher projections in your lab too then?
Yeah, we have a couple.
Okay. All right, good. So this will be a review for you guys then. So again, here's another example of a chiral carbon. It's got a methyl group, a hydrogen, a chlorine, and an ethyl group. These are all 4 different, therefore this carbon is chiral. This one is not, because it's got 3 hydrogens. This one's got 2 hydrogens, and this one's got 3 hydrogens. So these are not chiral, but this one here is. This is an example of one that is not chiral, because it's only got 3 groups attached to it. And so this is not a chiral atom or molecule. Oh, and that was a little bit of a quiz, and I gave you the answers. All right. You can have chirality in aromatic rings. Did you guys talk about that? Why is everything coming up backwards or whatever? So here is a dibromohexane. And this carbon here is chiral because it's attached to a bromine, a hydrogen, and then on this side, you might look at the ring and go, oh, well, this is all kind of the same, but it's really not. You've got, from this side, you go CH2, CHBr, CH2, CH2, CH2. But if you go this way, you got CH2, 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 CHBr, CH2. So those are different, and therefore this carbon is chiral. And a good way to look at this is to break the ring apart kind of in your head. And then you'll see that if you kind of imaginatively break this off, you see that this goes all the way around this way, and that bromine is on the second carbon out, but this one, or that was this side. This side here, if you break it out, the bromine is out on this carbon, and therefore these are not the same, and this is chiral. So that could be a bit tricky to see. Chirality is very important in biochemistry. It makes a difference, as we will see. So is this chiral? Is this carbon chiral? What do you guys think? You could just say it out loud or type it in there, whichever you want to do.
I'd say no.
You're
Yeah.
saying it's not. Anybody else? Yeah. Yes, this is chiral because you've got a hydrogen, a hydroxy group, a methyl group, and an ethyl group. So this is 4 different things that that carbon's bonded to. So this is chiral.
I thought it had to be like directly bonded like to the carbon. Like it's still chiral even if it's bonded to the hydrogen.
Well, remember, this is just shorthand. That's not the hydrogen is really up here. Oops.
Oh, okay.
Right.
Okay, yeah, I
This
thought the
bond
hydrogen.
here.
Okay, got it.
Yeah, this bond here is between the carbons, not the hydrogen. So,
Okay, got it. That's confusing.
all right, good. Good to clarify that. How about this one? Is this one chiral? You have to twist your brain on this one for a second. Okay, a couple people say no, everybody's saying no. All right, good, yeah. The reason why is this bromine's exactly in the middle. So this carbon has a hydrogen, it's got a bromine, and then now you got C, 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 Br, CC. But if you go this way, it's the exact same thing, C, 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 Br, CC. So this is not, did I draw it broken out? I guess I didn't. But again, if you break these out, you'll see that they're exactly the same because that position of that bromine makes a difference. Good. Okay, why is chirality important? When these things are chiral with each other, they are isomers, and they're called enantiomers. And the body will react to enantiomers differently. For example, right-handed epinephrine reacts 20 times more than left-handed epinephrine. Remember we talked about epinephrine last week. This is also called adrenaline. Your body will react with right-handed 20 times more than left-handed. I think I have a slide to show why. Most naturally occurring monosaccharides are right-handed. Consequently, left-handed sugars are not processed by the body, and that's what's often used in sugar substitutes. So instead of having glucose be D-glucose, a right glucose, they'll put a left glucose into the sugar substitute. It'll still taste sweet, but your body can't break it down and thereby gain weight from it. So that makes it a good sugar substitute. Amino acids, which we will get into in a few weeks, are the building blocks of proteins in our body, and they are almost always left-handed. Any questions there? Did you guys get into this kind of stuff in the lab, or was it just primarily focused on what is chirality? Just curious.
Just the what is.
Okay, so this is, now you're into new territory. Oh, I don't think I meant to delete this slide or revise it. I kind of liked this one better, but they're both there. So stereoisomers, this kind of shows you the categorization of this. Stereoisomers are isomers with the same connectivity, the same molecular formula, but they differ in their orientation of the atoms in space. What we're talking about tonight is enantiomers, which are stereoisomers that are not superposable on mirror images of each other. And here's an example down here. Diastereomers are stereoisomers that are not mirror images of each other, and our cis-trans isomers fall in this category. And you can also have stereoisomerism that results from a mirror image relationship at one or more chiral centers and have the same configuration at one of the chiral centers. So if you look down here, this is L-arabinose and L-xylose. They have the exact same molecular formula. So these are isomers of one another. But you'll see here that this one is a mirror image of that one. This one is a mirror image of that one. This one is not a mirror image of that one. So these are not enantiomers of one another because they're not exact mirror images of each other. But these are still isomers. They're called diastereomers. We'll get into this a little bit more. And by the way, I don't know if I said this in previous lectures or not, but the reason why cis and trans exist as stereoisomers is because of this double bond. Did I say that? If this was a single bond, you could freely rotate these things around and just make a match, and they would not be stereoisomers anymore. But with the double bond, you can't rotate these, and that's what makes these be stereoisomers of one another. And when it comes to stereoisomers, you can have a max, the maximum, and I bolded and underlined this because it's not given that there will always be this many, but the max is 2 to the nth power, where n is the number of chiral centers. So if you have 2 isomers that have 3 chiral centers in them, then the maximum stereoisomers you could have would be 2 to the 3rd power, which is going to be 8, right? 2 times 2 times 2 is 8. So there's a maximum of 8 stereoisomers you could have with 3 chiral carbons in them. If it was 4, then it would be 16 maximum stereoisomers. Some of them may be duplicates, so they wouldn't, that's why it's maximum that you can have on it. Okay. Any questions with any of that so far? Okay, so you guys have already kind of seen this. This is called a Fisher projection. And I think you guys said you did this in the lab as well. So here's our 3D molecule. You know, you've got a carbon here in the center and some different guys attached to them here. This is kind of how you would draw it with a stick figure like this, you know, with your little molecule toys. How that sometimes gets represented on paper is with wedges indicating that they're coming out of the page towards you, and dotted lines is that they're going back into the page away from you. So sometimes they get drawn this way. But a Fisher projection is where you draw it flat with the understanding that these guys are coming out towards you and these guys are going away from you. But this is a way of representing a molecule so that you can see chirality and handedness easier. And this was developed by a chemist by the name of Fisher. You said you did Fisher projections in class, right? So this is the one that we were just looking at a minute ago. This is a chiral carbon. Here's a 3D representation of it. If we take this and flatten it out into a Fisher projection, you've got this hydrogen coming out towards you and this hydroxy group going back away from you. And you've got the alcohol group here at the bottom. You've got your aldehyde group at the top. And you put a mirror in here. This is the mirror image of that. So here you have glyceraldehyde on both sides, but one is in the D configuration and one is in the L configuration. And I think I'm getting ahead of myself, but how you could figure that out is if you put this aldehyde group at the top, your main functional group at the top, and set it up this way. If the OH group is on the right, it's going to be D or right oriented. And if it's on the left, then it will be L for left oriented. I think we have a slide for this to show it more. So here's an example of 2,3,4 trihydroxybutanal. So here's our aldehyde group. That's why it's butanal, right? And we have two pairs here. We've got 4 possible isomers of this. So this first isomer A here has got the hydroxy groups both on the right. This one, B, has both hydroxy groups on the left. C has the top one on the left, this one on the right, and D is the opposite of that. This is on the right and this one's on the left. So there's 4 possible orientations of this molecule in the Fisher formation that we've drawn. A and B are enantiomers because they are mirror images of each other. You've got the OHs facing each other and the hydrogens are on the backside. So these are mirror images, and there's no way that you can superimpose this over onto this guy because these carbons right here are chiral, and so you're not going to be able to get these to superimpose. So they're enantiomers. These two, C and D, are also enantiomers because they're mirror images. So you see here we got H and H here with the OH in the back. Here the OHs are in the front and H's are in the back. And again, both of these carbons are still chiral. So you're not going to be able to superimpose D onto C in any way. So these two are enantiomers. But A is also an isomer with C, but it's a diastereomer with C because they're not mirror images of each other, but they still do have the exact same molecular formula. So A and C are going to be diastereomers. A and D are going to be diastereomers. B and C will be as well, and B and D will be as well. Any questions there? Did you guys do this in lab as well? Or no?
Yeah, we did this in lab.
Okay. So, and if 2 molecules only differ in one chiral center, they're called epimers. So determining handedness, or which way it'll rotate light. So we've got our same 4 isomers here. And again, you're going to start to the nearest carbonyl group. So that's your aldehyde group at the top here. And you're going to number down. And you're going to look at the highest numbered chiral carbon. So this carbon is chiral. This carbon is not because it's C double bond O. This carbon is not because you've got 2 hydrogens attached to it. These both are. So you look at the highest one. This is the from the top. So this is this carbon here. And so you look at the third one. If this OH group is on the right, then this is going to be a D isomer. It will rotate light to the right. And if it's on the left, then it's an L isomer. So this enantiomeric pair is, this A is D and B is L. And this pair here, C is D and D is L. Maybe I should have made these numbers or something because we're using a lot of letters here. But I think you get the idea. And you guys covered this in lab as well then. Okay. And here's an example I put in here because it's a ketone group. So again, you're going to order this with the ketone group at the top. And you're going to go count down. Here is a chiral carbon. That's your highest chiral carbon. And this is on the right. So that's going to be D fructose. And this one's on the left. So it's going to be L fructose. Okay, so is this one going to be a left or right isomer? Oh, I was going to put slide numbers on these things. I forgot. What do you guys think?
Right.
And why do you say right?
Because the top 2 are D. Right? 1, 2, 3. 1, 2, 3.
So is this carbon chiral?
Yeah, the last one is chiral.
So
and
that's
that would,
that's the one you look at. And this OH group is on the left.
but that's still clockwise. Isn't it? Oh,
It's
no.
it's just
You're
left or right. So yeah, so this is a left. So this is going to be a lever rotary or L, a left isomer. If this was over here, then it would be a right isomer. Okay. Are these two enantiomers, diastereomers, or neither? These two guys right here. All right, sounds like everybody's agreeing they're diastereomers. Why is that? You're correct. The reason why is they're not mirror images of each other. This first chiral carbon here is not a mirror image of that one. But they do have the exact same molecular formula. Remember, you got to keep that in mind, right? There was an extra carbon in this chain, then they're not isomers at all. Or if you had a bromine.
What would cause it to be neither?
Like what I'm saying, if you had an extra carbon over here, then these aren't isomers at all. They don't have the same molecular formula anymore. Or let's say you threw a bromine in here or something, then this now they don't have the same molecular formula anymore.
Thank you.
All right. I think that's the same as my other diagram. Why did I put it in there twice? I don't know. But there it is again. This actually looks a little better than the other one. Okay, enantiomers have the same chemical and physical properties because they are identical except for spatial alignment, but they differ in reaction to polarized light, like we said, their interaction with other chiral molecules and receptor sites in the body. D again means dextrorotary, which means rotate to the right. L comes from lever rotary, which means it'll rotate light to the counterclockwise or to the left. Polarimeters is a device that's used to measure the degree of the rotation. Did they have you use these in the lab at all? Did you get like sugar solutions and see how much it rotated the light or anything like that, or no? In lab, we learned for the isomers R or S. I have no idea what R or S is. R or S. Wow, that's strange. Maybe that's some kind of newfangled system that they've changed something. I'm not sure. But for purposes of this class, it's going to be D and L. I don't know what R and S is, to be honest. Oh, we already did this. What? Boy, I really got some extra ones. You know, a metaphor for this, for chirality, is a right-handed player and a left-handed player can use the same bat and ball because they are achiral, but not the same glove, because you got to have a right-handed glove and a red-handed glove. The thumb isn't going to be on the right place. In a similar manner, chiral molecules will not react with other chiral molecules in the same way. They're more like the baseball glove than the bat or ball. I thought that was a useful metaphor. So here's some examples. This is D-carvone, and you can see that it is in this configuration. It's in a dextrorotary configuration. If you split this out and looked at it, this is what gives you the taste of caraway seeds. But L-carvone, so it's in a lever rotary formation, gives you a spearmint flavor. So you can see that you go from, these are like very different flavors. You know, caraway and spearmint, I think, are very different from each other. And it's all just the same compound, just whether it's in a right or left formation. Epinephrine we talked about. So the receptor surface is designed in our bodies to for this to come in and come into these receptor sites and dampen pain, or this epinephrine doesn't dampen pain. Epinephrine boosts your energy, right? So if you look at epinephrine, they've drawn it so that the benzene ring with the two hydroxy groups is kind of this hexagonal blue structure. This is just to give you a conceptual rendering of this. The hydroxy group here is like a circle. The hydrogen is a hydrogen shape. And then this guy with this, the carbon and the nitrogen is kind of like the square shape. In the D configuration, all three of these points will connect to the receptor surface in your body. And this is why it's 20 times more reactive than the L configuration, because in the L configuration, you now have this hydroxy group up instead of down, and you're only getting this portion and this portion to be the correct match for the receptor in your body. And so this is why D-epinephrine is so much more effective than L-epinephrine. Does that make sense? It's just kind of a conceptual thing here to not get into all the chemistry of what is actually going on here. But the reality is these things are interacting chemically with these sites, these receptor sites in your body. But the OH group can't do that when it's in this vertical configuration like this. Any questions there? Okay, carbs in the body. So glucose, D-glucose, so dextrorotary glucose, is the most common in nature. It's the most important part and the most important one for the human body. This is also called dextrose because of the D for dextrorotary. This is the main component of blood sugar. So if you have elevated levels of D-glucose or dextrose in your body, then you're having high blood sugar. D-glucose is the primary source of energy for cells in our body. And in normal, healthy people, it's about 700 to 1,000 parts per million in your blood. Parts per million means 700 out of a million, or 1,000 out of a million, right? You guys familiar with parts per million? So 700 to 1,000 is considered a healthy range. If you get above 1,000, then that's where you get hyperglycemic. And if you get below 700, you're hypoglycemic. And here's the structure of D-glucose. So again, it's a D because this highest chiral carbon, the OH, is on the right-handed side. And so this is really the workhorse in the human body. D-galactose. So galactose has 1, 2, 3, 4, 5, 6 carbons in the chain. It's got an aldehyde group on one end, and you've got your hydroxy groups with an alcohol on the other end. It is in a D configuration because this is chiral. I'm not sure why this is highlighted. This is chiral and this is to the right. So this is D-galactose. D-galactose is a component of lactose, which is breast milk. It is also a sugar that powers the brain. It's used in processes in your brain. It's also present in markers that designate blood type. So if you are of type O blood type, you have a red blood cell here that is attached to a galactose, an N-acetyl glucosamine here, and then a galactose, and then a fucose. So these monosaccharides chained together, attached to this red blood cell, is what determines that you're of an O blood type. And so this is like a oligosaccharide chain attached to that blood cell. Here, if you have a type A blood type, you have a red blood cell that has a galactose, an N-acetylglucosamine, another galactose connected to a fucose and an N-acetylgalactosamine. This determines an A blood type. I'm not going to test you on this. There's no questions on the test about that, but just so you kind of see that this is how these things are used in our body chemistry. And then type B has a different configuration as well. I don't really know why they call them A, B, and O. Does anybody know? You guys probably got more biology background than I do.
The O is a mistake. It's supposed to be a 0. That's the only thing I remember.
Oh, really? Interesting. Yeah, I can't ever remember. I think I'm O. What's the like universal donor? Is it O negative?
O negative is universal.
Negative.
Yeah, I'm O negative. I'm Mr. Take my blood. I think my wife was A positive. They used to do blood tests when you got married. But I don't think they do it anymore. I think they just automatically give pregnant women shots to deal with the trouble that can come about from being positive and negative. But anyway, D-galactose, major factor in the body. D-ribose. So over here we have ribose, which is made up of 1, 2, 3, 4, 5 carbons with it's an aldose because you've got your aldehyde group up here. It's on the right, so it's D. D-ribose is a major component of RNA, and 2-deoxy-D-ribose is the main component of DNA. So here you see the oxygen's gone from this one here. This oxygen is gone. It's now 2 hydrogens. So this carbon is no longer chiral. It's deoxidized, so it's a deoxy-D-ribose. This is a component of DNA. Monosaccharides in ring form. Like we said, they exist in straight chains, but they're typically more stable in a ring formation. So here's a Fisher projection of D-galactose, our favorite buddy. We've looked at it a few times. Got 6 carbons in there. This is how it would look like in a ring formation. And did you guys do Haworth projections in lab? You talk about that at all?
I don't believe so.
I don't remember that.
Oh, okay.
No.
So this is another way of trying to render what a ring would look like in 3 dimensions. So this part of the ring is coming out towards you, and this part of the ring is going back away from you. So kind of think of it as the ring is like flat, it's horizontal. So there's a front edge and a back edge to it. And then now these OH groups and hydrogens are going to be either up or down depending on how this is formed together. So if you just take D-galactose and start to put it in a ring formation, this is how you get this. This is named after a chemist by the name of Haworth who developed this. And what happens is how you go from a chain to a ring is that it bends over upon itself. As we said, these 2 ends are going to combine together. And then what happens is this carbon rotates. This bond right here rotates. So you see here we've got H, OH down, and this is straight. This rotates so that the OH group is now facing that aldehyde group. Your CH2 is now in the up position, and this hydrogen is in the down position. So this is basically just kind of rotated around. You guys see that? And now this hydroxy group can react with this aldehyde group to close the ring. And the oxygen forms a bond with this number 1 carbon on the aldehyde group. And so you get this bond forming over here with this. But now you've got 1, 2, 3, 4, 5 bonds on this carbon. So this bond has to break in order to do that. And then you end up with this becoming an alcohol instead of a carbonyl group. So that's the step process in it. So let's go through that again. So you've got the straight chain. It starts to bend on itself like this. Then this 5th carbon bond rotates so that the OH group is now closer to this aldehyde group. This then forms a bond with the carbon, popping that double bond with the oxygen over here. And then that closes it off and gives us this oxygen is now closing that ring in with the carbon here. And you now have a hydroxy group here instead of a carbonyl group. And this H is moving over to here. But this is still galactose. It's just in a ring formation. Any questions about that process? So in the case of D-galactose, it's D because this highest chiral carbon has the OH group to the right. So this is D-galactose. And when you form the ring, your keep clicking that by accident. Your substituents on the right side in a Fisher projection, quit clicking, are going to be facing down, and those that are on the left-hand side will be facing up when this ring is closed off. And so you now have basically this oxygen in the ring. So this is a heterocyclic ring now, because it's not all carbons, there's an oxygen in that ring. When you look at the OH group on this side of the oxygen and this chain, this alcohol group on this side, if they're both up in the plane of this ring, they're both above the plane, this is called a beta isomer. It has the same elevation around the oxygen. So it's both of these are up, it's the same elevation. If it forms the ring so that one of them is down and one of them is up around this oxygen, then it's called an alpha isomer. And so you can have beta D-galactose, that's this one here, and you can also have alpha D-galactose, that's this one here. So they're both dextrorotary, but one can be in an up position and the other one can be in a down position. Any questions there? Did you guys cover this in lab at all, or we're just totally out of your lab zone now?
I feel like we're pretty out of the lab at this point.
Okay. I need to look at those labs and see what's going on in there just so I have an idea. Any questions there though?
Which one is this one on the slide? I'm not finding it on my end, on my notes.
You mean you printed these off?
Yes.
Oh, I don't think I added this today or anything, but maybe you printed it off before I.
Is there like a header on it? Like, for example, like some of them say.
Yeah, well, it's going to be
pros and
right after
cons.
this slide. I didn't put a header on it, but it's right after this final slide with showing how the ring closes.
Okay.
I have a question. What determines whether or not it's a beta or alpha? I understand that if the isomer is up or down, but would they not always do the same thing? Or am I missing something?
No, I mean, the way chemicals are, these reactions occur, it just depends. There may be a favored configuration. I don't think we get into that in this class, but it could be that naturally that this might be the, don't take this as a fact, but I'm just hypothetically saying here, it could be that the beta is like 75% of the time and the alpha is 25% of the time, or something like that. But both of these can form when this guy closes in on itself. So it can happen either way.
So for our purposes, just know that the beta is isomer up and alpha is isomer down.
Yep, that's the best way to, it's around this oxygen. You got to look at that oxygen in the ring and look at whether they're up or down, or they're the same side or opposite sides. I don't know if so Haworth projections, these ring 3D attempts to draw like a 3D ring thing. They're used to demonstrate the spatial structure of cyclic monosaccharides. And then, like we said, on the alpha, the functional groups around the oxygen are at opposite elevations. So this is the alpha form. You have this OH down here and this alcohol above. And the beta is going to be where they're both have the same elevation around this oxygen. But both of these are identical to this Fisher projection. So they're the same molecule again, it's just in different configurations. This is an example of monos, which is another aldose here. D configuration, the functional group on the highest carbon is above the ring. In an L configuration, it will be below the ring. So you look at that highest carbon from the original, this guy right here, if you put this at the top and look at your highest one, then if it forms above the ring, that's telling you it's a D isomer, and if it's below the ring, it's an L isomer. Probably should have put a sample problem in here. But you can see here, this is above the ring. So that's going to be in the D configuration, which is, so this is D monos. And so this is going to be beta D monos, and this is going to be alpha D monos. Okay, if we do a weak oxidization of a monosaccharide, so if you use some kind of a weak oxidizing agent, you will cause this aldehyde group here to oxidize into a carboxylic acid group. So you've added an oxygen in here. So you go from COH to COOH. And this is called, in this example, this is D-glucose. and this becomes D-gluconic acid. Gluconic based on the fact that it's glucose. So you can see here how this is building on stuff we've covered previously. We talked about weakly oxidizing aldehydes into carboxylic acids, and now we're seeing it happen in a sugar in your body or your food or whatever. If you strongly oxidize D-glucose, you will oxidize both ends of this. So this aldehyde group will go to a carboxylic acid group, but this alcohol group will also go to a carboxylic acid group. And this is called D-glucaric acid. And what is this? This is on your test for this weekend. What would you also call this? This is a dicarboxylic, right? You've got two carboxylic acid groups in here. So again, we're kind of building on that previous material. So a strong oxidizing agent, a weak one will favor this carbonyl group up here. Remember, this is C double bond O with an H also attached to that carbon. So a weak oxidizing agent will preferentially go after that aldehyde. But if you have a strong oxidizing agent, it will attack both the aldehyde and the alcohol at either end. Enzymes in the body can also have an effect where they just attack the alcohol end. And so an enzymatic reaction of D-glucose can just oxidize your alcohol into a carboxylic acid, leave the aldehyde group alone. And this is referred to as D-glucuronic acid. Again, I don't think I'm going to make you name this stuff anywhere, but knowing that an enzyme does this, whereas a strong oxidizing agent goes after both of these groups, and a weak one is only going to attach the aldehyde. That's the key things to understand here. You can also reduce a monosaccharide. So here again, we have our friend D-glucose. In the presence of hydrogen and some catalyst, what you will do is you'll convert this aldehyde group into an alcohol. It gets reduced because that carbonyl group, that double bond O, is going to pop and become an OH. And this now is a dialcohol. You've got alcohol on either end. And so this gets called D-glucitol. This compound is actually has a common name of D-sorbitol. And this is used in gum, chewing gum, because the bacteria cannot react with this like it can here. This glucose powers our bodies. Well, it also powers bacteria. But if it's not glucose, it's glucitol, then that bacteria can't react with that and cause tooth decay. And so this is often a component that's added to modern chewing gum to reduce tooth decay. As a matter of fact, the dentist told my wife a few years ago that chewing modern chewing gum actually prevents tooth decay. It becomes like a preventative treatment because it helps your mouth, the bacteria in your mouth not attack your teeth, because this acts as an inhibiting agent in your mouth. So this is a reduction because we're adding a hydrogen to this aldehyde group on the end. Okay, I threw this one in. So if you printed this off before Sunday, This may not be in your deck. You might have to go back and print this off, but I added a few in, just trying to refine my slide decks. And I want to do just a quick review. So if you remember, we talked about aldehydes reacting with alcohol will form hemiacetals. And if you react a hemiacetal with alcohol again, you will get an acetal. So a hemiacetal has an ether group here, and it has a hydroxy group here. An acetal is two ether groups like this. And that becomes important because beta D-glucose acts as a hemiacetal. So this is a beta D. This highest carbon is above the plane, so it's a D-glucose. I've got this problem with my fingers. I keep accidentally clicking stuff all the time. And it's in a beta configuration because both of these are above the ring around that oxygen. So this is beta D-glucose, but this is also a hemiacetal. You've got this OR group here. You've got an O connected to an R chain and an R chain over here. But you also have this alcohol here. And so this is a hemiacetal. It can react with an alcohol in the presence of acid and become an acetal. And what you do is you end up replacing this hydrogen, this OH group, and this hydrogen pop off to give you water. And this methyl group connects into that oxygen, producing another OR group here. And this is called methyl beta D glucoside. Again, don't worry about this name, but the methyl here referring to this methyl group that's now in here. And of course, beta D is still, because it's in a beta D configuration. So this is forming what's called a glucoside. Glucosides are used in skincare products because it removes dirt and oils by breaking surface tension on your skin. So it helps break up that surface tension, and the dirt and the oils won't go into your pores as easily. So you're taking glucose and turning it into glucoside, and that gets used in skincare products. I always tell people, so I think I told you guys the first night, they asked me to teach this class a year ago, and I was like, I've never had biochemistry. Did I tell you guys this? Lydia said way out about something. So I was learning this biochemistry last year in preparation for teaching this, and this is amazing. This is like a whole level of chemistry that I never learned before, and I just find it very fascinating and really interesting. Wait till we get into more of the body chemistry. It's really cool. Okay, remember we said phosphate esters are important in biochemistry. That was one of my sample questions for you because we had talked about that in chapter 5. So here's an example of this. So we have our friend beta D glucose here. This can react inside your body with phosphoric acid. Yes, phosphoric acid can exist in your body. It's just very, very dilute. But it reacts with this to form a beta D glucose 1 phosphate. So you pop off again this hydrogen in this hydroxy group and get water. And this phosphate connects into that oxygen there. I'm sorry, this hydroxy group pops off and this hydrogen to form water. This phosphate then connects to this oxygen. And this is now a phosphate made from glucose. And this is a component that is very important in our bodies metabolizing carbohydrates. So this is like an intermediate step in a process for our bodies breaking down carbohydrates. We'll get more into this later. Amino sugar formation. So here we have a sugar. This is one of our monosaccharides. I don't remember which one. Is it the same as, it looks like it's a alpha configuration. This is an alpha D-glucose, it looks like to me. Yeah. In our bodies, this can get converted to what's called an amino sugar. So this is still a sugar, but instead of, one of this hydroxy groups gets replaced with an amino group by reacting with some amine compound. And so this goes to from D-glucose to D-glucosamine. And glucosamines are used in our body in the formation of cartilage. So cartilage is like in your ears, it's in your knees, it's in your joints, places like that. Glucosamine is a component of that cartilage in your body. And that's called an amino sugar because the sugar has an amine group in here. So Lydia, what were you saying way out about one of the slides before?
Yeah, you had asked about our labs and whether this was something familiar or if it was way out of our
Oh,
territory.
I see. Oh, okay. I thought you were saying one of these slides was like way out, using like 70s language, you know.
No.
That's way out, man. Okay. All right. So that's monosaccharides. We now look at disaccharides. These are also important in body chemistry. So disaccharides are formed by two monosaccharides reacting with each other. One will act as a hemiacetal, and the other one will act as an alcohol, and they will combine to form a disaccharide. These are also called glycosides, and you'll get water as a byproduct. So here we have an example where this OH group, so this is acting as an alcohol, so this OH group is going to pop off, and this hydrogen is going to pop off from this monosaccharide acting as a hemiacetal and give you water. So here's our H2O over here. And then now this oxygen is going to link with that carbon. And this is called a alpha linkage. An alpha linkage because this is in the alpha orientation. So this bridge here between these two, this is a disaccharide now, and it's linked between the two here with this oxygen, and this is called an alpha linkage. There's also a beta linkage that we will see in a minute. Like maybe now. So here we have beta D-glucose. So this is in the beta formation because both this and this are on the same elevation around that oxygen. Again, this is going to act as a hemiacetal. This glucose is going to act as a alcohol. We're going to pop off our water, just like we said here. And then this guy, though, now is linking from a vertical position above the ring to this guy, this carbon here. And this is called a beta linkage. It often gets drawn like this rather than a V shape to try to represent that. And both of these are called a 1,4 linkage because you're linking with the number 1 carbon on this ring to the number 4 carbon on this ring. So this is called a beta 1,4 linkage. This is an alpha 1,4 linkage. This, by the way, this compound right here is called cellobiose. It is a disaccharide that is an intermediate step in the formation of cellulose, which is a polymer. And we'll see this in a minute. Any questions there? Okay. Another example here of a disaccharide is maltose. Maltose is also called malt sugar, and maltose is formed when starches break down. This can happen inside of plants. It can also happen inside of the human body. If you eat lots of starches, those starches get broken down into sugars in your body, and one of those products is maltose. This is a common ingredient in baby foods. And you can see here that this is alpha maltose, and you can also have a beta maltose, where they have a different configuration, where this one has got the OH group on the opposite of the plane from this alcohol group. So this is an alpha maltose, and these are on the same elevation above the plane. So this is a beta maltose, but both of these are alpha linkages. I know that can get kind of confusing. Again, I don't think I test you a whole lot on this type of thing, but just being aware of it. Lactose is another sugar. This is the main ingredient in breast milk, for example, whether it's human or animal. Alpha lactose is sweeter than beta lactose. So here is alpha lactose, where you have the OH group on the opposite side of the ring of the alcohol. This is beta lactose, where they are on the same elevation. And these are beta linkages here that it's showing. Beta lactose forms an ice cream as it sets and ages. It goes from alpha to beta lactose. And this is what gives ice cream its gritty texture if it gets too old. You ever pull out old ice cream and get kind of that grittiness in it? That's because that sweeter alpha lactose is converted to beta lactose as it's sat there. Sucrose is common table sugar. This is a disaccharide as well. Sucrose is a glucose monosaccharide connected to a fructose. So this disaccharide is called sucrose. That's kind of a common name for it. All right, any questions on disaccharides before we move to polysaccharides? We're good to go. So again, our polymer is you have a repeating unit over and over again, and this can happen with monosaccharides. You can form long chains of monosaccharides with that repeating monosaccharide over and over again. If they're all the same, so if these were all like glucoses, then this would be called a homopolysaccharide. If it's got multiple monosaccharides in the polymer, then it's called a heteropolysaccharide. This is an example of starch. Starch is also referred to as amylose. And what you see here is you have, keep clicking again. You have, this is a homopolysaccharide because these are all the same. And you've got this chain made with these alpha 1,4 linkages between each monosaccharide. And this can go on and on and on and form very long chains of sugars. This is starch. This is why starches are they're good for you in moderation, because it does give you these sugars that provide energy to your body. But lots of starches, you're going to end up with a lot of sugar in your body. And then what your body does is it says, holy cow, I can't deal with all this sugar. And it starts storing it as fat. And this is where eating a lot of starches can cause you to gain weight. And I don't remember if it's in this deck or a future week where we talk about that. It might be here. Oh, it's here. Yeah, so your body stores excess glucose as glycogen. Glycogen is a multi-branch polysaccharide of glucose. So here we have two glucoses connected together, but they can branch and connect to another chain. And then this chain could branch and connect to another chain, so forth and so on. So these are multi-branched polysaccharides, and that's called glycogen. And it's how your body stores glucose. It's much more efficient to store this as glycogen. And then what happens is, what we'll see is that your body will peel off glucose as it needs energy. If you exercise quickly, you like suddenly do a burst of exercise, then your body dips into these glycogen resources to provide energy. It's like, oh my gosh, he's doing something. He's running like crazy. He needs energy. And so it'll dip into these glycogen reserves and start peeling off glucoses to give you energy. And so this is why aerobic exercise is very good at burning body fat, is it's going to dip into these reserves and consume this stuff. If you're just strength conditioning, that will do it to some extent, but it's not like that accelerated aerobic exercising. Carbohydrate loading is something that athletes do, like runners will do this before a race to maximize their energy levels. So any of you guys runners? Any of you guys do half marathons or marathons or anything? I don't. I only run if some guy's chasing me with a knife, and maybe not even then. I might take my chances. But runners will sometimes do what's called carb loading so that they maximize their glycogen in their body, so that as they're doing a big race or whatever, they're going to have extra glucose in there to power your body. Okay. One of you is training for a half marathon in April. Cool. Any questions there? We'll come back and see this again. Cellulose is a linkage of monosaccharides. These are beta linkages in long chains. And they form linear structures with these beta linkages. Linear structure better than branching for structures. Yeah, so if you have branching, like here's amylose. This is what a starch will look like in its formation. It will sometimes curl in upon itself a little bit. These little blue dots are your monosaccharides in the polymer chain. Amylopectin is a starch that's branched. Sometimes starches will form branch things like this. But you can see these aren't going to provide as much structure because you've got all these empty spaces in here and that kind of a thing. Cellulose gives you this very linear structure with these beta linkages, and it therefore will form a structure more like this. And this is cellulose is what gives the rigid structure in plants. So your weed stalks and your trees and all that have a lot of cellulose in them, and that gives them that hard structure. And here's an example of glycogen. That's your multi-branched stuff that your body is storing that in. Cotton is 95% cellulose. So cotton is almost all cellulose. Wood, interestingly, is only about 50%. Okay, humans and most animals cannot digest cellulose. Animals such as cows have special enzymes in their stomachs that break it down. So cows are one animal that can digest cellulose. We can't digest. If you eat a tree, you're not going to break that cellulose down. It's just going to run through your system. But dietary fibers are healthy for you to eat because they help move food through your intestinal tract. They will also make you feel fuller with fewer calories. And this aids in healthy digestion. So dietary fiber is actually good for you, even though your body's not breaking it down. Dietary fibers also will bind with cholesterol and pull it out of your body. So people with high cholesterol tend to not have very high fiber diets. So the more non-sugar coated fibers that you can eat will help cut down your cholesterol. People with high fiber diets are not often obese. Many diet products have high cellulose because they make you feel full faster, and your body can't digest them. So they're empty calories, they just go right through you. Americans, unfortunately, eat about half or a third of the recommended amounts of dietary fiber. And I'm guilty of that too. I'm very guilty of that. So you want to have more fiber in your diet. Chitin is another example of a polysaccharide that's made out of amino derivatives of glucose. So we've got these glucoses here, but they've got this amino group attached to a carbonyl group attached to a methyl group. But you've got these beta linkages in between these as well. These polysaccharides are called chitin, and it's what is used to give structure to shells, animals with exoskeletons, and even structure to fungi have chitin. I didn't know that until I started teaching this class. I knew about shells and exoskeletons, you know, like crabs or whatever. Ants, I think, have exoskeletons. I didn't know about fungi, so fungi have chitin in them. Okay, acidic polysaccharides. I think we're getting close to the end. Yeah, I've got 4 slides left. So acidic polysaccharides are heteropolysaccharides that contain an amino sugar and a sugar with a carboxyl or sulfate group. Please tell me I have a diagram. Okay, oops. Okay, an example of this is hyaluronic acid. So here we have this amino group with a carbonyl group in it as well. They're chained together with these beta linkages. Here and here and here. This is another way sometimes beta linkages are rendered to show you that you've got a vertical elevation bonding with a horizontal or down orientation. So you've got beta linkages here of these substituted monosaccharides that have a carboxylic acid group on it and also an ammonium carbonyl group in here as well. This polysaccharide in particular is used in the body for fluid in the eye and a lubricant in joints. So it tends to be viscous and aid in like making like what do you call that? Like greasing something up, lubricant, right? These are like natural lubricants in your body. And because of this, it's often called the goo molecule. So, you know, if you pop your eyeball, you're going to get goo out. This is the major component of that. Heparin is a chemical that is an anticoagulant. A lot of elderly people are on heparin to keep them from forming blood clots. And it binds to the protein involved in blood clotting. And here you've got this carboxyl group up here, but you've got a sulfate group down here. And again, in this case, it's an alpha linkage between these. And these form long chains as well. And this is heparin. So again, I'm not going to test you on draw the diagram of heparin or anything like that. But knowing that heparin is an anticoagulant is useful to know. And then finally, dietary carbs, kind of coming back to that. Dietary carbs are made up of simple carbohydrates, mono and disaccharides. These are often referred to as sugars. Also complex carbohydrates. These are polysaccharides or starches. Again, these dietary carbs come in rice, potatoes, wheat, corn, and these things. They also could come from fruits and vegetables. It is recommended that 60% of your diet be carbohydrates. Refined sugars, though, are called empty calories because you get no nutrients out of those other than just the energy from the sugar. So if you eat refined sugar, it will just give you energy boost, right? That's where kids like eat too much sugar, they get hyper. But they're totally empty because all it is energy. There's no nutritional value other than that. Whereas your other carbohydrates will break down and give you other nutrients that your body needs. And you guys have probably seen a food pyramid before. Your base is water, fruits and vegetables and breads, meat and cheeses should be lesser, and then your stuff that contains a lot of refined sugar and fats should be a very minimal part of your diet. Unfortunately, Americans are kind of almost the inverse. Americans tend to be chronically dehydrated, and they tend to eat a lot more junk food than these healthier things down here. And I think that brings us to the end. I've got problems for both 4th and 7th edition here for you to look at and try your hand at. And it is 5 minutes to 9. So again, test this weekend. It will start at noon on Friday, run till midnight Sunday. Remember, we got a little bit of a downtime early Sunday morning. It's on chapters 5 and 6. So study for that to get ready for that test. But if you do have time, or you can make time, go over this and study it so that you can come back, I guess, in 2 weeks. So you got all next week to look at this carb stuff again. So you got a little bit of a buffer there. You know, come back 2 weeks from now, we'll review carbohydrates. And if you've looked this over and kind of studied it more, then you'll be able to come to class with questions or anything like that. Okay, any questions, comments, concerns, anything? Everybody's fried. Oh, Lydia said that she did some half marathons too. Cool. Probably nothing I'll ever do. Probably should, but
You have a second to look at those problem questions with me in the 6th edition book.
yep,
Thank you.
yep. Have a good night, everyone. You can take off if you want.
Thank you.
Okay. So which chapter you want to look at? Like chapter 17?
Yeah, I just yeah, let's see
Flip back there.
what's the title of the, I don't have my, I just put my notes away.
It should be amines and amides.
Okay.
Is that chapter 17 in your book?
Oh, perfect. It is chapter 17. Yeah. And then the other one that we're getting question or
Well, let's look at a couple
Or
problems.
examine.
Let's look at a couple problems in
Okay.
chapter 17. So go to the back of chapter
Okay.
17.
Yep.
My 17.1 says contrast nitrogen, oxygen, and carbon atoms in terms of the number of covalent bonds.
Give me 1 second. I'm almost there.
Oh, okay, sorry.
On the chapter. No. That's good. Thank you for taking the time to look at this. Okay, what does yours say?
So 17.1 says contrast nitrogen, oxygen, and carbon atoms in terms of the number of covalent bonds they usually form in organic compounds.
Perfect. Yep. And then 17.2 says contrast nitrogen, oxygen, and carbon atoms in terms of the number of non-bonding electron pairs they normally pose as present in organic compounds. So it looks like we have the right.
How about number 7? 17.7 says indicate whether or not each of the following compounds contains an amine functional group.
Yeah, and then the A is CH3, then CH, CH3, and then off of the CH is NH2
Okay,
for
good.
A.
Maybe we'll just pop over and check a
Is
later
that what you have
one.
for one of the answers?
Yep. So let's look at another one just to make sure we're looking good. It looks like it.
Okay.
How about number 71, 1771? It says classify each of the following salts as an amine salt or quaternary ammonium salt.
That was number 71. You
Yeah.
said?
Yeah.
Could you repeat it?
Classify each of the following salts as an amine salt or a quaternary ammonium salt.
I might have. Well, that looks like it's 17.73. Is that right? Because
No,
I think it's
I'm looking
okay.
at 71.
Okay.
My 73 says indicate whether or not each of the salts in 71 can be converted back to its apparent amine through reaction with a strong base.
Okay, that one might be different.
So these sound like these might be a little different.
Yeah.
Well, I think what you can do though is you can look at the slides and then you can pick problems out that match the material in the slide. You know what I'm saying?
Right.
So if you see a problem that's talking about something and you look at that and go, I don't even know what the heck they're talking about, that's probably not one that I would normally give you.
Okay.
Um,
I know I mean, what am I trying to say? So I obviously want to pass your class, but chemistry is not my strongest suit in life, but I'm trying. I've been like borderline with my exams this far. I just want to keep it there, or a little bit of an improvement, just because, I just don't want my grade to drop.
Sure. Yeah, so just try to pick out the problems that match the subjects that are in the slides, and I think you should be okay.
Okay.
Don't get bogged down into questions that don't look like they match anything in the slides.
Right.
The
Okay,
good news is the biochemistry side that we're now in, it gets a little bit more conceptual. It's not as heavy duty. So you might find that you do better in the biochem side. But you want to try to get a good grade this weekend.
Yeah,
for sure. But I'm just,
definitely.
as encouragement, it gets more conceptual and less like nitty gritty chemistry type stuff. Just like you saw tonight, this carb stuff was much more, I kind of view it as like a higher level chemistry. We're not getting into the nitty gritty of how these reactions happen and all that kind of stuff, right?
Right.
So
could we look at the other chapter real quick, please?
I should have stopped this from recording.